As the verse in our Rig Veda says, Ano Bhadra Kratvo Yantu Vishwatah. Let the noble thoughts come to us from every side. With the verse, let's welcome our first invited speaker, Professor Eswaran. The title of his talk is Anti-Cancer Active Water-Soluble Nanocarbon Materials. Sir, please.
It is indeed a great honor and a privilege to be present here. I address the chair for the session, Professor Sushant K. Banerjee, Dr. Shakil Ali, and Professor Indupal Kaur. And I thank the organizers for inviting me to give this talk. I'm not going to waste my time, get down to business, and I'll keep the non-essentials to the minimum. That's my name. Idea is that we should be able to communicate with each other. Those who are present here, those who will hear this talk subsequently in other media. If you see here in the very first slide, what we have shown is we have made 3 water-soluble, nano, carbon-based anti-cancer drugs. In vitro testing tells us that they are very effective to the extent of around 90% ability to kill different cancer cell lines. And what is most remarkable, it appears that the cytotoxicity is very, very minimal. We have heard many speakers in the morning emphasizing this point that there are so many unbelievable side effects for cancer drugs, which are not only prohibitively costly. We have shown 3. Can we have the pointer? See on the left, you see one, Let me go closer to the per gram. This is only 4,000 rupees per gram. Only 4,000 rupees, the last one. But we are on the verge of getting graphene. We are on the verge of getting graphene from the India Innovation Centre for Graphene in Kalamassery, Kerala, under the Ministry of Electronics and Information Technology. I am not emphasizing on the characterization full, but we got very good idea about the molecule from its molecular weight, precise up to the 4th decimal place, not possible for the other 2. Absolutely homogeneous, that's what the pharmaceutical industry wants. These nanotubes are vertical with tremendous electrical and magnetic properties predicted for them. All of them are fluorescent. Why do I pick up electron spin resonance spectrum or electron paramagnetic resonance spectrum and fluorescence? Because these are going to be useful for following these molecules in living systems. So this is the first part, but as I conclude this part, I must say these compounds are also candidates for theranostics. To define the word theranostics, therapy plus diagnostics. Not only you diagnose, but also you cure the patient. Yes, we have to develop that, it's a long way. I am here claiming only one thing. We have in vitro results, but our path is long. So I look for collaboration. Money that is required for the next future steps, very hard and very long journey, very costly. We have already spent a lot of money doing this work. Please help me with this. So I think he should manage this rather than me. Yes, these are the 3. This is fluoroacidamine. Can you notice a slight pinkish color? Completely water soluble. The one on top here, that is somewhat grayish in color. It's almost white. We have a 2-step strategy for making these things. The nanocarbon, which is insoluble, we thermalize with our azide, an aromatic azide we make in our laboratory. These are green reagents made in small quantities at a time. The starting material, the nanocarbons, costly. But This two-step synthesis published previously in Journal of Biomedical Nanotechnology 2020, again 2022. This I have already referred to, but the exact molecular weight of the fullerene-based molecule is 1492.99120. The pharmaceutical industry would love such a compound which is reproducible and entirely homogeneous. I thank Professor Prasenjit from the Regional Center for Prasenjit Kuchayat, my collaborator from the Regional Center for Biotechnology. If he is present, I would acknowledge his contribution and help. All this work which I am covering in this slide is from his laboratory. The U937 cell lines, CT26 colorectal carcinoma cell line, these have been subjected to the analysis using the immunoblot, and it shows that these compounds are bringing about very efficient apoptosis at concentrations similar to the concentration of doxorubicin, a very currently commonly used drug, with terrible side effects, particularly I understand cardiotoxicity. The percentage survivability of the C26 cell line in Professor Prasenjit's lab is shown. We were granted this patent on 31st January 2023. Publication happened independent of this. The process was long from 2016-17. Yep. This is the second paper in the series, Journal of Biomedical Nanotechnology 2022. The patent was applied first and the paper sent for publication. Immunoblot shows the increased expression of pro-apoptotic molecules like BAX, caspase-9, and cytochrome C in U937 cell line, understand is monocytic cell line. In this paper, the densitometry has also been done by the student. In addition to that, in 4T1 breast cancer cell line, similar results were obtained. So also in CT26. What is the main point in this paper? That these compounds are showing very good anti-cancer activity with little or no cytotoxicity. Having done this lab work in an academic laboratory, it is absolutely necessary for me to move to a higher level, which meant much more money being invested. So we approached, among others, we approached Reliance Life Sciences in Mumbai, and they did the work in 2 reports. This slide summarizes our results from the first report submitted to us around the month of August 2023. If you notice, the yellow line in the figure is below the red line here. The yellow line shows that the RGO, reduced graphene oxide manganese 2 complex, there is the presence of a manganese atom which came during the oxidation step, which was the second step, which was carried out to make the molecule more polar, therefore water soluble. And water solubility is not only important for processability, but one must note, for toxicity also, these nanocarbon materials become harmless when they are totally water soluble. The work in Reliance was done on the HeLa cell and the cytotoxicity studies on L929 cell line. The bars are showing the anti-cancer activity, and what we are claiming based on the report from Reliance is that 90% activity, anti-cancer activity at 125 microgram per milliliter. This is in the first report. And the cell viability at 90%. In the case of RGO, RGO stands for reduced graphene oxide manganese 2 complex, 90% anti-cancer activity at 250 microgram per milliliter, while preserving more than 70% cell viability at that concentration. This table summarizes the 3 water-soluble, nano, non-cytotoxic compounds I am presenting here. In the middle, On the right is fluorescein. If you notice, as given at the concentration of 250 micrograms, the anti-cancer activity is 95%. Cytotoxicity is low. In the case of reduced graphene oxide manganese 2 complex, similar 95.760 activity and almost no cytotoxicity. Unbelievable. Single-walled carbon nanotube, there is some toxicity, but the anti-cancer activity is very good. These are independently tested by Reliance Life Sciences Mumbai. This is a result we got just last week, fortunately from the same lab, second report, which has just come to us. We have done tests with them in 5 cell lines. These include the A549, the triple negative breast cancer cell line, MBAMB, and the FADU, and MCF7. What are the results? Summarized by us, the A549 lung cancer cell line, in the case of fullerene or fluorescein, named by me is 87, single-walled carbon nanotube complex 80%, and Arjyo complex 77. As we go to the next cell line, MCF7, I presume, 75, 89, 78. These are in comparison to doxorubicin being taken as a standard 100%. The next one, what are we looking for? The results here are quite remarkable. If you see 86, 94, and 90%. That means in lung cancer, human breast cancer, triple negative breast cancer cell line, and for that matter, a huge range of cancer cell lines which we have tested. The results are very, very superior. As I try to close my talk, against the triple negatives, cancer, breast cancer cell line, MDAMB, at 500 micrograms, doxorubicin used as a comparator. Single-walled carbon nanotube based material showed 75% inhibition. Fluorescein based on fullerene gave 89% inhibition, and reduced graphene oxide manganese complex showed 78%. What do I look for? I look for to having graphene from the new center set up by the central government, Kerala government, and Tatas, so that we can convert the wonder material, graphene, also into water-soluble variety of the same kind, and we'll test it for anti-cancer activity. This is the summary and the road ahead. What is the summary? We have characterized 3 water-soluble nanocarbon materials, fully characterized, published, patented. These show very high anti-cancer activity compared to doxorubicin, with little or no cytotoxicity, 250 micrograms per milliliter. They are showing activity against lung cancer cell line, human breast cancer cell line, and even the triple negative breast cancer cell line. What's my prayer? I am not here to advertise. I seek support from the entire community. And as I wrote in the Journal of Biomedical Nanotechnology paper in 2020, the editor did not cut my sentence. I wrote, we wish that these results be tested the world over and they found to be true. We are prepared to provide this technology on a no profit, no loss basis, so that it reaches the worldwide community and the people who need that support. Thank you for your patience. If there are any questions, I'm available for some more time. My students are available and we can continue. Thank you so much.